Welcome back to the deep dive. Today we're jumping into a paper that really presents a huge, almost counterintuitive breakthrough in single-cell biology.
It really is.
I mean, usually you have to choose, right? You either get high-throughput sequencing, millions of cells, or you get precise spatial context, knowing exactly where that cell came from.
It's always been a trade-off.
But this new stereo cell platform, it just completely smashes that barrier. It delivers high throughput sequencing while, keeping all the spatial coordinates. And it integrates multi-omics right on a single chip.
Yeah, and it's a remarkable piece of engineering, really motivated by the limitations of current tech. Your traditional droplet methods like 10x chromium, they physically discard the cell's location.
They just throw it away?
Gone. And they also really struggle with large or fragile cell types. So StereoCell addresses this by using these existing high-density DNA nanoball arrays, DNBs. These are nanoscale capture spots, maybe 220 nanometers in diameter, and they just act as these universal sort of encapsulation-free landing pads for intact cells. You just drop the cells right on top.
So let's get to the first big question. A totally new architecture like this, does it compromise the data quality? I mean, the researchers went right for it. They benchmarked StereoCell against 10x using human immune cells.
And the answer was a very clear no. Actually, the opposite. StereoCell generated higher transcript counts per cell overall. But I think more critically, it delivered results that were just far more accurate.
More accurate than the standard.
Way more accurate, especially when compared to the flow cytometry gold standard. For example, when they looked at monocytes, the 10x data showed this significant capture bias. It reported proportions that were almost 12 percentage points higher than the biological reference.
Wow, that's a big error.
That's huge. StereoCell, on the other hand, it captured proportions almost identically to the reference. It's just a much more unbiased profile.
Okay, that's a massive improvement for common cells. But, this platform is being sold as revolutionary. So how does it handle the things current tech just avoids, like complex tissues, cell interactions, or really, really big cells?
Well, that's where the planar format of the chip is so clever. It lets them integrate all these different data layers at the same time.
Right, which brings us to the multi-omics part, the StereoCell CITE. They combined the RNA sequencing with, what was it, 154 oligo-barcoded antibodies?
Exactly, targeting surface proteins. And integrating that protein data with the transcriptome was, well, it was the key to resolving this really fine-grained cellular heterogeneity.
So you could see things you couldn't with RNA alone.
You absolutely could. They were able to distinguish specific T-cell subclusters, like these CD112 plus T cells, that were completely invisible in just the RNA data. It's the difference between knowing a cell's type and, knowing its precise functional state.
And the flexibility here is just wild. They went way beyond common immune cells. They tackled these complex, oversized samples that microfluidic methods just can't handle.
Like skeletal myofibers.
Right, these huge multinucleated structures. That's not like sequencing a single bead. That's like trying to sequence a rope.
And this is where that spatial context really shines. They captured different segments of the myofibers that corresponded to specific functional areas. They could actually map the markers for the myotendinous junction and the neuromuscular junction on the same fiber.
And then they went even bigger. They sequenced mouse oocytes, the largest cells in mammals.
Yeah, and they didn't just sequence them. They identified gene modules with distinct subcellular localization patterns. They could literally see where specific RNA transcripts were living inside the cell.
Which they then validated with imaging, right?
They did. With SMRNA-Amplify, it confirmed the whole thing.
So this ability to profile these incredibly complex and large structures while keeping spatial context down to the subcellular level, that feels like it pushes this technology toward actual clinical application.
I think so. You can imagine sequencing precious tiny biopsy samples or finding rare circulating tumor cells. The high throughput makes it possible, and the spatial fidelity, well, that opens the door to investigating dynamic changes in huge cells, like cardiomyocytes, heart muscle cells, at a resolution we've just never had before.
So to recap our deep dive here, there are maybe 3 core takeaways. First, StereoCell gives you unbiased, high-throughput single-cell RNA profiling, and it's actually more accurate than the established methods.
Second, it integrates multi-omics RNA, protein, and imaging on one chip, which lets you see phenotypic diversity that's invisible to RNA alone.
And third, the platform can capture these huge, structurally complex samples and even lets you detect subcellular RNA localization.
We should probably note the one key limitation. Because it's an encapsulation-free design, you can get some minimal lateral RNA diffusion. A little bit of contamination between cells that are right next to each other, they'll need to keep working on mitigating that.
A fair point. But here's a final thought to leave you with. Considering StereoCell can already pick out gene expression modules linked to structures that look like extracellular vesicles, or EVs. What might we learn about the microenvironment and how cells talk to each other if we start routinely mapping these critical non-cellular components in complex 3D tissue models? Something to think about.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1. To help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2. To build a supportive community where we can exchange the most valuable experience-based research advice. 3. To share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.